So I run an organisation called EcoHealth Alliance, and what we've been doing for the past decade or so is really trying to focus on fusing the disciplines of ecology with the health sciences to create a new discipline which is being called disease ecology or eco-health, and in other terminology, One Health. And I want to try and show today, I'm going to talk a little bit about some of the debates over the impacts or potential impacts of climate change on infectious diseases and other health issues, but really to give a couple of case studies of work that I've been involved in where we got the opposite result to that which I hoped for, and which I think explains the complexity of this, of bringing together different disciplines. So first of all, infectious diseases are a fundamental part of human history, and as you go back through history, you see these repeated cycles of effectively new emerging diseases coming into our population, wiping out large portions of it, and then becoming endemic. And you can go back through history, and we can start maybe at the biblical plagues, where there's really good evidence that we were forming cities of the right size to allow animal diseases to persist in our population and cause repeated outbreaks. So there is perhaps some truth to those myths. The great examples of the Black Death in Europe that probably wiped out a quarter or more of the European population at the time. Smallpox, a disease that Westerners took over to another country and effectively caused population extinctions in some parts of Latin America of groups that were naive to these infections, diseases that we'd become used to in our endemic population. But recently there's been this resurgent interest in the process of how diseases pop up and cause problems in a group of diseases we call emerging. Really that goes back to Legionnaire's disease and the emergence of HIV/AIDS, and more recently SARS and H1N1 influenza. I'm going to talk a little bit about that later on. But the important thing to note here is that in almost every case of a new disease appearing in our population and beginning to spread, the drivers, the causes of that disease's emergence are effectively ecological issues we can deal with population models, ecological models, and ecological approaches. So a good example of that, if we think of what is now a human disease that doesn't affect wildlife as far as we know, measles virus, and we look at this phylogenetic tree for measles, you see the most common recent ancestor is one shared with rinderpest, which is a cattle disease. If you look at a molecular clock for that, the probable spillover of that pathogen from cattle happened at about the time we're forming big cities and first domesticating cattle. So it's an ecological process, and we're repeating that over and over again as we reach into new environments and do different things to the environment. One of the, you can sort of summarize it in a conceptual framework of there are different groups of populations. There's wildlife, which harbor lots of new viruses and unknown pathogens that have not yet emerged. There's livestock, where we have a very close relationship with, and there are our own populations. And we've fundamentally meshed together on the planet now through globalization and the livestock trade and our relationship through anthropogenic change to the environment with wildlife. The factors that cause diseases to emerge across all of these systems are very similar, and they're all related to things we do, to the environment, to our own demography, that cause changes in the dynamics of populations of pathogens. Now, it's fairly complicated, and I'm sort of adding today climate complexity. I'm adding to that the complexity of dynamic populations of wildlife and their pathogens. But the important thing is that these are fundamentally predictable things, because we know about how we're changing the environment. We know about how pathogens change in wildlife populations. So we're beginning to get to a situation now where we can predict the outcomes of our changes to the world. Now, a lot's been talked in the literature about climate change and its likely health impacts. And here are a few examples of hypothesized, proposed impacts of climate change. Things that are very closely linked to climate, to weather, to longer-term climate issues, and things that have been hypothesized by a number of authors with sometimes really good data as linked to recent changes in climate. And these run the gamut from non-infectious disease impacts like heat stress, things related to air pollution, fairly obvious, through to diseases that are transmitted by vectors that rely on water to reproduce. So obviously as climate affects water tables and water surfaces and temperature, it increases the ability of those pathogens to be transmitted, through to more difficult to understand relationships as we talk about wildlife pathogens that spill over into people. And this is a huge issue. You know, there's lots of glory to be had in the literature, publishing in the rival journal Nature as well as in Science, on these links and trying to form these correlational links. But it's often hugely controversial. So this was a nice review in Nature that sort of summed up the health impacts and pointed out that, you know, really focused a bit on the more obvious health impacts, the health impacts related to water quality, to air quality, and pretty believable and with some good data to back it up. But then we have more difficult to test and to correlate issues, like the movement of the vector for blue tongue disease. This is a virus that affects sheep in Europe, and it never before has moved into the Mediterranean region. It's now endemic there. It even went as far as my home country, my original home country, England. And for a disease that's driven by a vector to go to miserable cold England, something's going wrong. But there's been a lot of talk about this being linked to climate change, but again, very little actual correlational data to support that. A huge debate in the literature about malaria, especially highland malaria, because obviously as we look at the impacts of climate change, we're going to see an increased impact on an altitudinal gradient. And vectors of malaria, mosquitoes, respond very nicely to temperature changes in the lab, so you can measure how a 1 degree change in temperature will affect production of eggs, the biting rate, the reproduction of the malaria parasite inside the mosquito, and therefore develop a nice model that predicts the impact. And lots of really good papers have been published on this. This is a prediction for Zimbabwe, and this is the baseline in 2000 for malaria suitability for the vectors and the disease, and this is what it's going to look like in 2050. So there's lots of doom and gloom in the literature based on very good models. But the problem is there are also lots of papers that say this is purely correlational and other things are happening out there too. For instance, there's been a huge upsurge in drug-resistant malaria. So as you look at incidence of malaria in humans, in some places that's increased because of the increase in drug-resistant malaria. So trying to decouple those two factors, climate change and the spread of drug-resistant strains, is very difficult and has led to lots of interesting and amusing debates between viciously opposed teams of scientists. But I think my take home is that something is going on, and we're going to begin to see, as we get longer term data sets, really significant evidence for climate change as a driver of increased disease risk to people. And this is a good example. Really nice evidence that some of the El Nino events increase the amount of hospitalizations for diarrhea in various parts of the world. But what I want to talk about today is to neatly avoid that controversial issue and to talk about two case studies that I've been involved in looking at disease emergence, because if emerging diseases are intricately linked to the changes we make to our demography and to ecology, and climate change is one of those changes to the environment, then surely they're also driving disease emergence. Can we test that? Can we look at some examples of emerging diseases and see if climate change is involved in that? And that's exactly what we set out to do a few years ago with a number of emerging diseases, including ones that affect people and others that affect wildlife. And I'm going to talk about two examples of those now. But first of all, what is an emerging disease? An emerging infectious disease can be very clearly defined as diseases that have recently spread, moved into new areas, recently moved from wildlife or livestock into our population, expanded their geographic range, or increased in virulence due to some factor like resistance to antimicrobial drugs. And there are lots of important examples of those. The reason why we focus on emerging diseases is because of their impact. If you look at HIV/AIDS, it's now 40+ million people affected. SARS didn't kill many people, but the threat of SARS is a rapidly moving pandemic that emerges with no therapy, no drugs, no vaccines at the time it emerges. And that's why there's such a scare around H1N1, so-called swine flu, because the threat of pandemic emergence is high. without the vaccines and drugs to deal with it. So, as a wildlife biologist and parasitologist, I was very interested in a series of new viruses emerging from bats. Now, it seemed really odd to me. I was working at CDC at the time when this disease, Nipah virus, emerged, the one that I've circled there. It's a disease that emerged in Malaysia from fruit bats, flying foxes, beautiful animals, pretty rare, they're out there in the environment. Doesn't seem to be much contact with people, so why should a new disease spill over from bats into people? And furthermore, why should there be so many pretty nasty diseases that seem to originate in bats? And this is what we set out to investigate. What is the cause of the underlying driver of emergence for Nipah virus? This virus emerged in Malaysia in 1998-99, 265 patients, 105 fatalities, so it's a very lethal disease, 40+% case fatality rate. It's been emerging repeatedly in Bangladesh for the past decade, and the case fatality rates are even higher over there, upwards of 60, 70, sometimes 90% of the people infected die from this disease, and there's no vaccine and there are no therapies, no drugs. So this seemed like an important disease to me. In Bangladesh, it's begun to be transmitted from one person to another, so it has the potential to evolve into the next pandemic, the next HIV or SARS. So how do you investigate a disease and try to understand what caused its emergence? And to us, this is a collaborative effort. So we need veterinarians to understand how the disease spreads between one animal and another. We need medics to do the work on the ground. We need epidemiologists to go and interview patients, understand what they do that puts them at risk for infection. And we need ecologists to write mathematical models to describe that system so we can begin to test hypotheses. Now, the way we approached it is to go back to the original farm where Nipah virus first emerged. This virus came from bats and got into pig populations in Malaysia, which then cycled the virus very rapidly and then spread it to people. And pig farmers were the only people originally infected by this disease, but it was traced back to fruit bats by CDC. The problem is, normally when we intervene in a disease, that's where we leave it. We don't move any further than just trying to stop transmission. No one was interested in really trying to understand what caused it to emerge so we can stop the next one. Now, when you go to Malaysia, you'll notice straight away if you step out of the plane at the wrong time of year, you get this horrific smell of burning in the air. And there's someone referred to it earlier, the Kalimantan forest burning, creating a haze event over Malaysia every year. The reason for that is huge deforestation in other parts of the greater Malaysian region, Kalimantan, Sumatra. And these are some of the data on the amount of forest that's being chopped down annually, and then fires that are set to clear the land for agriculture. So I was very interested in this. And we began to formulate a hypothesis that maybe what happened here, this is the outbreak site. This is where the outbreak was, in a village called Songhai Nipah. By the way, if you ever go to Malaysia, these are great sites to go for a sightseeing trip, down to the places where these viruses emerged. Also, there's some great ones in England too that I'll recommend later, some of the plague villages. But the original spillover event, which is what we wanted to see, why did this virus first get into humans, was in a place called Ipoh here, where there are big pig farms. That's up here. This is a picture taken at the height of one of these big haze events, and you can see these are the origins of the fires, and the prevailing wind tends to blow it over to Malaysia and cause an extreme haze event. Now, bats are notoriously sensitive to smoke. They'll fly away, and these bats can fly long distances. The wingspans are a meter or more in diameter, and they're big bats, and they'll fly for 100 kilometers without stopping. So one idea was, well, maybe the virus originated in Sumatra, in bats here, which then flew over to Malaysia to avoid this haze event. It's an interesting idea. So we started to see if there's any evidence for that. Well, first of all, we thought it was cool because we're not climate scientists, but we could pretend to be proper scientists and have NASA as a logo on our talks, which I thought was great. So this is some data from NASA. It's not really data, it's just a nice picture of this haze event in 1997. So remember that date, October the 1st, 97. We also thought that, this seems to be something that's taken seriously in literature. This series of wildfires has been linked to other biological phenomena and published in important journals, so there must be something in it. And not only that, when we looked at the data on the first human cases, this was the big outbreak in Nipah, but about 2 or even 3 years before, the first few cases where the virus actually spilled over into the first human was right when this spike of particulate matter was in the air. So it suggests that maybe this haze event is part of the story. We looked at rainfall, and round about the time of that haze event, it was that big El Nino in the late 90s, so the area dried out. When an area dries out, it reduces the pollination of fruit trees that these fruit bats need to eat. And you can see that has a significant output on oil palm production during the next quarter. So there are some serious biological effects that we can see with data from these fires. But what we wanted to know was whether the bats actually fly to Indonesia and back anyway. Is this just a natural part of their circulation? And do they already have this virus in their population? So this is where it becomes less sciencey and more difficult. Now, one thing we noticed straight away, we started to see a problem because we found new cases of Nipah in people. We found that some of the workers were infected earlier than we thought, which precedes the El Nino event. So it starts to throw a spanner, a wrench in the works of that argument. Anyway, so back to our bats. We wanted to know where these bats cycle. So the first thing we had to do was climb trees and catch bats, which is easier than it sounds. And by the way, that's not me up in the tree there. I was down at the bottom of the tree sipping gin and tonic. We actually hired a British guy who was crazy enough to climb. He still works for us. But you have to climb right to the top of these trees, string nets between them, and each individual bat that you catch, you have to winch it down, catch it, not get bitten, anesthetize it, bleed it, and release it later on. It's a lot of hassle. And you have to, sometimes you get worried about whether you're going to catch the pathogen or not. But we don't have huge security issues. It's a pretty rare virus. But you do have to remember that sometimes when you work in the mangroves, the tide might come in and completely wash away your field site. But this is good work, and it's good fun, and it's quite interesting. What we found was there was a whole new technology of satellite collars that you can attach to wildlife and set them free and watch where they fly and just download the data onto your laptop and observe them moving from A to B. So the test we wanted to know was, do bats fly between Malaysia and Indonesia outside of an El Nino? And this was the first bat we caught. He was called Colossus, and he was huge, about this big. And what he did was, superbly, flew exactly where we didn't want him to go. We had we caught bats all over Malaysia and repeatedly found movement between the two regions and across political boundaries. These are migrating animals that move following fruiting of tropical trees, especially durian, which is disgusting, but they really like it. For those of you who had it in the room, know it's a horrible, horrible thing. So basically, this completely threw out our El Nino hypothesis. Bats routinely move between these two areas outside of El Nino. The virus has been there a long time, as evidenced by serology studies that we did, and the first cases were prior to the initial El Nino event. So we moved on to our second hypothesis, that maybe it's just simply the production of pigs, which was novel in Malaysia, in a more intensive way, was the driver. And what we find is that if you look at just data on mango production and pig production, it's an absolute dual system all across Asia. Even tiny farms with 5 or 6 head of pigs will have one mango tree or one durian tree. And it's just something that in Asian cultures you do. You have pigs, you have some fruiting trees, and you have a pond with carp in it. So what happened is, this was when Nipah emerged and the government closed down some of the larger pig farms. So you get a rapid drop-off in pig production, but you also get a simultaneous drop-off in mango production. So these systems are working together. The mangoes attract the bats, and the pigs are susceptible hosts. So we went back in great detail and looked at the original farm where this virus emerged, and this is now 3 or 4 years on. Found the owner, interviewed lots of people, and gathered lots of data. We looked at the first few human cases, and what we wanted to know was why there was such a long time between the first case and then 2.5 years later, the big outbreak. How could a virus persist in a population for that long? We gathered huge amounts of data, and this now goes to the interdisciplinary side of it to truly understand why this disease emerged, and created a mathematical model that very accurately describes the transmission of this pathogen within a pig farm, and trying to understand what happens if you have a farm of a certain size. You go over a threshold at which it becomes endemic, it becomes a huge problem, and allows the disease to emerge. And that's exactly what happens. What we find is that if we imagine this virus getting introduced into the pig farm just once, it always dies out. There was no way, given the size of farms in Malaysia, that the virus persists. So just look at the green graph. They're infectious pigs in our model. If you introduce the virus once, you get this big boost of infection. A lot of animals die, some become resistant or recover, but the virus always dies out. The only way to allow this virus to persist for that long period of time that we saw is to repeatedly introduce it to a pig farm of greater than 20,000 head of pigs. So what we have is an increase in production intensity in Malaysia, coupled with planting fruit trees around the farm that allowed this virus to emerge. It probably wasn't anything to do with El Nino in the end. But this is really good because it allows us to, first of all, we've tested in a proper scientific way the hypotheses for a disease emerging for the first time. It's really hard to do. And then produced a model that we can use to advise the Malaysian government where they can put pig farms if they want to recreate that industry. So that's one example where we thought it was going to be climate change, but it wasn't. Now to an example where we thought it shouldn't be, but it was. And this is a wildlife disease of amphibians. And the reason why we worked on this is that in the past few decades, there's been this huge global decline of amphibians since the 1970s. And really what's been important about this is that they've been declining and going extinct in areas that are protected, where they shouldn't really have disappeared. Lots of ideas were put out about this. Was it climate change? Were they toxins, stress, UV light? But this is what it looks like. When you do field studies and repeated surveys, you get this simultaneous crash in population to zero across a site from multiple species. So to us, it looked like a disease, and specifically a bit like Cortez introducing influenza to the Latin American, native Latin Americans. Introducing a pathogen to naive populations, you get this rapid die-off to zero. Eventually we found a disease, and it looks like we did Koch's postulates, we showed that it was highly infectious, and this disease we found repeatedly at every site where amphibians had declined. It's called chytridiomycosis. It's a fungal disease of the skin of frogs. It lives only on keratinaceous cells on the surface. And it's probably the most important cause of so-called enigmatic declines of amphibians over the past 10 years. Now, when we first published that, there was a huge debate on whether a disease could wipe out its host. It didn't make sense to people. But what happens here is you've got lots of different species. Some survive, but the fact that the pathogen survives in the surviving frog means that all the other species can be driven to extinction. And to prove the point, we set up a study, and we were funded by NSF through an interdisciplinary research group, IRCEB, to bring together a huge group of different disciplines and look at this issue. One of the things that we did, and this was under Jim Collins' tutelage, was with Karen Lips to go down to areas in Latin America where the disease hadn't got to yet and wait for it to spread, with the idea we believed this was a disease that was spreading naturally through the population. And that's exactly what Karen Lips did. Year after year she went down, surveyed the population, found no infected animals, healthy populations. Then suddenly the disease hits and the whole population basically falls off a cliff. Brilliant proof of concept, but bad news for the amphibians. So again, can we go to the underlying cause and say what's driving this disease to emerge? And we had two hypotheses, and again, strong debate and disagreement between the groups. The first, which was our group, we believed it was an introduced disease, just like smallpox to the Aztecs, just like huff and mouth disease to the UK. Humans have spread this disease around the planet, and it's getting into naive populations and wiping them out. The other group suggested it was a disease that's found everywhere, but something's changed in the environment and made amphibians more susceptible, namely climate change, either UVB or some other issue related to climate change, or pollution or some other factor. So we pursued doggedly this introduced disease hypothesis and found really good evidence for a global trade in frogs for the pet trade, for the pet industry, for the food trade. By the way, if you eat frog's legs in DC, you're eating bullfrogs, so you really shouldn't do that. It's pretty disgusting. Not only that, the bullfrogs bred in Latin America or Asia, North American bullfrogs taken over there, bred in farms and brought back live with the pathogen. So it's not a good thing. And this is one of the farms. We actually went out to farms across Asia and across Latin America, found evidence of the disease, documented its spread in the trade, so we could try and get some policy change on this. This one family frog farm probably had about 800,000 live frogs at the time we went there. And it's just a mass of frogs. It's pretty horrible. Full of disease, completely open to the environment, and they're an alien species that invade. So it's a perfect system for a pathogen to get around. And this is a very systematic, very well laid out farm in Brazil. These little cement drums contain a few hundred bullfrogs, which is great, but they're completely open, and then the water's sluiced out into the local river system, so the pathogens spread beautifully. And the pet trade is huge. 5 million live amphibians a year into the US, 1 million live bullfrogs so we can eat them and show off to our friends. So we tried to find evidence and do something about it, and we put a policy forum out in Science. And by the way, AAAS members in the room, those policy forums really do make an impact. The OIE, which is the World Trade Organization allied body that makes a decision on whether trade should be blocked due to diseases, have made this disease notifiable. So the US has signed up to it. That means now there's a way to stop the spread of that disease, which is really, really important, really nice. But at the same time we're doing this work, the climate change debate continued. Climate change could still be driving this disease. And we started finding some weird data too. There's a site in North Carolina, South Carolina, called Savannah River. And this is a famous site for amphibian research. It's the longest continuously running drift fence in the world. And it's shown that there's very little declines down there, not like the ones you see in the tropics. Yet when we looked at some museum specimens, we found evidence of this pathogen. So how could this pathogen be there and not cause declines? What's going on? So we started looking at it and worked with Whit Gibbons down there and found that actually there are declines. If you look at this species, it's declining. Now this species here is declining. And what we did is we linked it to every time we worked out the exact requirements of that species for standing water to breed. And we found that basically the whole area is drying out in that long-term drought over the 90s and causing a rapid decline in amphibian populations, irrespective of disease. So amphibians respond to climate very well because they need water to breed and they're very dependent on water. So there may be something to this climate change hypothesis. This is Monteverde, where Alan Pounds has been doing the work, which is the background to a lot of this for a long time. This is the cloud forest where the golden toad used to live, which is now extinct, probably due to this pathogen. And what he showed really nice in a series of papers is that if you look at the temperature fluctuations over long term at those sites and the number of species of amphibians you can see, they respond exactly to that temperature fluctuation. Then if you look at declines, the vast majority, 83% of declines, occurred after a warming period and a drying of that area. But the debate continued until, we started looking at really high mountain sites with some climate change scientists from Lamont Doherty who work at the Gloria site in Peru. This is Anton Simon's work. So what you can see here is over the 1931 to 2005, retreat of a tropical glacier and the opening up of a bridge from one valley to another. Now, we hypothesized that if you open up a bridge like that, amphibians will be able to migrate up the mountainside and connect with other populations. And sure enough, we did the field work, we went down there, and that's exactly what we find. In these ponds which are formed where glaciers used to be, they're now absolutely chock full of larvae of various amphibian species, full of this pathogen. So there's strong evidence there that as climate changes and warms up a mountainside, amphibians invade from the lowlands and take the disease with them. And this might explain why there's a climate change link to this pathogen. Now, can we take this a step further and really start to come up with some broad-scale approaches to predicting the big threat to our populations, our own human populations, these emerging diseases? If we look at a series, this was where we were in 2001. This is Tony Fauci's work from NIAID, just listing out where emerging diseases seem to originate and trying to see a pattern. So what we thought we could do is, can we do this better? Can we use ecological approaches to try and understand the patterns of disease emergence by looking at what causes them to emerge, which are ecological issues, and specifically focus on those diseases that originate in wildlife, which are, by the way, the big ones: HIV, SARS, Ebola, all the influenzas. So trying to focus on those biggest threats to public health and use ecological approaches. So we started to do a simple, large-scale database of emerging diseases. It took a long time, actually. It took a couple of years. And this is what we got, which is every emerging disease for the past 4 decades, with the point origin, as far as we could get it from the literature, for every known emerging pathogen. There's about 400 or 500 of them. And if you look at these data, the raw data, straight away you realise that where I come from, England, is pretty horrible place for pathogens. And in fact, where I live now, New York, is equally bad. But of course, I swiftly correct myself, these data are completely biased, because what they also include are places where people are looking for pathogens. The richer the country, the more hospitals, the more chance of finding a new emerging disease. So we're probably under-reporting from parts of the Amazon, parts of Central Africa, parts of Asia. So we corrected for that by looking at the origins of studies published in Journal of Infectious Disease every year for the past 3 decades, about 17,000 authors. Geogridded them and ran those with our analyses to try and correlate disease emergence with various biological factors. And what we're trying to do is to say, does the presence or absence of a disease emerging in every 1 degree grid correlate with various factors like human population density, rainfall, latitude, very importantly mammal diversity, and then our correction for bias. Now, the problem we have in trying to predict future disease emergence is we don't know how many pathogens there are out there. There could be 1,000, there could be 1 million, there could be 100 million. If you start to look at wildlife and do some viral discovery, you very rapidly find new pathogens that are previously unknown. So the way around that is to use mammal diversity as a proxy for pathogen diversity. So what we assume is that every mammal has a roughly equal number of native pathogens, and then we look where the diversity is and say that equals roughly the risk of a new pathogen, or the presence of a likely new pathogen. We use human population density really as a proxy for the ecological changes that we do to the environment, the anthropogenic changes that drive disease. And that's what we try to understand with this analysis. So by splitting the data into 4 different groups, you can start to see patterns. So what we find is for zoonotic diseases from wildlife, this is probably the most important finding, that there's a strong correlation between wildlife diversity, the source of new pathogens, and human density. So it's pretty much what we thought would happen. But because we've got spatially gridded data, we can then produce a predictive map of where the next new pathogens are going to come from. So this is the first ever, we published it in 2008 in Nature, the first predictive map of hotspots for the next SARS, the next influenza, the next Ebola. And what we see now is instead of the UK and New York being the hotspots, we start to see areas of rapidly developing tropics where biodiversity is high and there's lots of human activity becoming the hotspots of where the next disease will emerge. And it's interesting to note that where the next big disease did emerge, which was H1N1, is predicted on this map as a hotspot. So while everyone was looking in Southeast Asia, Indonesia, this is a hotspot for the next disease. So when you do this sort of work, and you're really doing it on a shoestring, we were funded by a couple of NSF efforts. One was Human and Social Dynamics, which was really nice. And that support was critical, just exploratory grants. And you publish a paper in Nature, you really want to try and make an impact. And I think we did, because a couple of years later, USAID used that analysis to look at disease instead of as a biosecurity threat to the US, but more as a development issue. If the underlying drivers of disease emergence are things that humans do to the environments in the tropics, then let's spend our money more wisely and focus some of it on those areas and do advanced surveillance in the areas of the tropics where things are happening to stop diseases emerging. So that's what we're now working on with a bunch of other organisations and focusing on these hotspot regions to do viral discovery to try and identify potential zoonoses before they emerge. And it's a big job. And you know, when you're able to hire a bunch of bright postdocs in the lab of different disciplines, you get great science, you also get great acronyms, and this was a good one, I thought. This is actually Andy Dobson's idea, Rita, you'll appreciate that. Instead of Wikipedia, we'll have Sikipedia. And it's not launched yet, but it's currently behind the scenes, where we've got expert review of the first origins of every emerging disease known. We've been able to really refine the hotspots from this, which is our original publication, to this really detailed predictive maps. So you can go into a country and decide where you're going to do surveillance to test out the model. So you can look at the red areas to find the new pathogens, but you can also look at the green areas to test aspects of the model. Is it really accurate? Are those green areas cool spots for emerging diseases? Now, part of the work we're doing now is to focus down in more detail on, because that was pretty preliminary, on a few drivers of disease emergence. One is land use changes. This is data from the Institute of Medicine's review of emerging diseases, the most significant drivers. So we're looking at agricultural industry changes, climate and weather, and land use changes. I just want to show you a little bit of what we're doing with some of the climate work now. So going back to those bat viruses, the idea here is that bats are very mobile. They'll respond rapidly to people planting durian in a new region when the climate warms enough for them to do that. So they're going to be good exploiters of a changed climate. But bats happen to carry these pathogens. So does that then mean that those pathogens are going to move into new areas and cause new disease outbreaks? So we focus specifically on Nipah virus and Hendra virus, the related sister virus, and the reservoirs for that. So this is the distribution of all the bats that carry those diseases. And it includes outbreaks that happen regularly in Australia. We're using a sort of standard ecological niche models specifically used before to understand bat dynamics based on temperature and other issues. So, you know, just looking at the maximum, minimum precipitation, temperature where bats have been reported recently, and then trying to model that and sort of predict where they should be, their distribution, and then predict where it's going to be in the future. We have great data from GBIF and other places on where bats have been found for this group of bats, and we have an excellent collaborators working on climate change using 19, I think, different factors, different ranges for climate factors that drive disease spread potentially and drive the change in reservoir distribution. Using an ensemble model and MaxSense software, and using a pretty, I think that's a moderate IPCC scenario. And what we find is that we start to see where not only the current distribution could be, but also where it will be likely in the next few decades for the bats that carry this disease. And that's what we've found for this group of viruses. Now this virus, there's evidence of this virus in Africa, but what we begin to see is, you know, a huge expansion of the range of those reservoirs. And if you do a sort of change map of current distribution to predicted distribution, you start to see areas where perhaps we should be focusing our surveillance to see if that virus is emerging in bat populations there. And we've already seen it in India, we know it's in Africa, so potentially there's a larger distribution. Now, you can even extend this globally. There are bats that are very similar in parts of Latin America. No one's ever described this virus there. So we're doing some surveillance in Brazil and Mexico and other parts of Latin America, specifically testing for those viruses that we know are found in other parts of the world that we wouldn't normally have done without this sort of modelling. So we've been able to convince people it's worth testing for those new pathogens. So this is the first trial of what we're going to do for, basically, if we can do it, all viruses and pathogens carried by mammal hosts, using ecological niche models for the hosts that carry those pathogens, and hopefully getting a better way to predict where these pathogens are going to emerge. Now, I really like this sort of work, and it's fundamentally it's interdisciplinary. You know, you can tell from what I'm saying, I'm not a climate model, I don't understand it very well, but I understand it enough to know when it's good and when it needs to be improved and how we can better improve it. And I think there's a great future in that niche between the disciplines. I think that's where the good science is going to be, and that's where the good applied science is going to be too. I'll just show an example of applied science that I think is really cool. As you're all flying home from this meeting, we've been able to predict, we've got a paper submitted now, using the data from our global hotspots for emerging disease, plus a huge amount of travel data into and out of airports, and local economic data at how good countries are at stopping diseases. We can now tell you which are the most risky airports on the planet for emerging diseases. And I'm happy to say we've still got a few in these places, but you can start to see these areas in the tropics where wildlife carry new pathogens are going to be the sources of future pandemics. But we're also incredibly vulnerable here because of this global travel network. The first place those diseases are going to go to is part of that travel network, the places that travel the most, which is Europe and the US. So that's the good news at the end. I want to say one thing. The funding for this, really, the work we've done is now being funded largely by USAID, but originally came out of a series of grants, which we still have, with an amazing program called the Ecology of Infectious Diseases program. Now, I think there are a few, and only very few, programs that originate in NSF and where they reach out to NIH. And we heard before about social science envy of natural scientists' budgets and ability to bring in money. Well, of course, NSF has envy of NIH's budget, which I believe, Rita, is probably an order of magnitude higher. There's a huge amount of money goes to diseases which don't kill many people, which aren't as significant as the next pandemic. And I think that the vision behind that program was fantastic, and it's still an extremely strong program, one of the very few examples of good interdisciplinary funding of research. I also want to mention, you're going to hear a lot more about this group, Diversitas, which is a part of the ICSU family. We have an EcoHealth cross-cutting network within it, and Hal's going to tell you a lot more about that later on. Thanks very much.